Well, welcome back. We're in Bio 211. We are looking at Chapter 4, and what we're doing is we're finishing epithelia. Before we get going, again, I just want to make sure people understand how to use these types of sort of videos. And that is, when you've read your textbook, you probably paid attention inside the lecture. Maybe even I'm getting ahead with this video, and that's completely fine. It's to help you focus on, when it's really comes down to crunch time, what are the important things to be thinking about. And one way to help you with that is, here's some questions from part 1. So you've read the book, you watched my last video, you're going to see that I have a series of questions for you here. There's only 4 of them, but these represent what I think are really important ideas. So before we finish up the epithelial stuff, let's think about going backwards. What does it mean? In all of the tissues inside of the body, they arise from 3 progenitor types of tissues. What were their names? What does it mean to actually arise from one of these 3 types of tissues? If you think about the outline that I drew for you, remember it looked something like this. Let me see if I can do this for you. Boom. Right? Remember that cartoon there. What does that actually mean? And, where did the tissue types, if the excitable tissues are up here, right, what did they arise from? Okay, that makes sense. Cool. Number 2, 4 types of tissue. Where do you find them? I mean, where do you find each of the connective tissues, the muscular tissues, the neuronal tissues, the epithelial tissues? This is sort of like a trick question, because in reality, you should be able to find all of them everywhere. I'm not going to answer the next 2 for you, but what are the different junctions that hold cells together? I'm looking at that cartoon where we went from essentially the basolateral surface. We looked at the lateral surfaces first, and we worked our way to the basement membrane. What are the proteins that are used to build junctions that hold cells not only together, but also to some sort of basement membrane so they don't move anyway, or away from one another? And then finally, if we think about epithelia specifically, what are the different ways that Mother Nature assembles them so they can actually be functional? So you should be able to answer these questions. Nudge, nudge, hint, hint, wink, wink. And when you think about a class quiz or something like that, these are the ones, or exam, or for that matter, final exam, you'll see that these questions, maybe not literally like I have them for you right here, will be the ones you're going to see on the exams. Okay, so let's finish up. Columnar, you saw this in the previous video, where essentially they're taller and wider, and they're specialized for secretion and absorption. And remember, I said the transitional ones, these are the special ones, because an organ, say like the bladder or the ureter, it actually has to change its size, and you have to be able to withstand these pressures and these forces. And what happens, of course, is it can literally transition for a flat type cell. And by flat, we mean let's say columnar epithelia that has a flat top that can be squished down to cuboidal. Think about what happened if you lined a balloon and you filled with water, right? What would happen to the epithelial cells inside of that? When we think about what this looks like, here are those columnar epithelium. They might have some modifications on them where they are, in this case, ciliated. And remember, cilia from chapter 3 move materials in a unidirectional manner. And here inside the trachea, so there's the trachea right there, and there's the thyroid gland right there. These are lined in such a way that when you're thinking about something that irritates your throat, Mother Nature has to be able to remove it from your body. So materials catch. Mucosal cells make a mucus, it sits on top of this, you catch it and you move it out. And, if you think about what an expirant is doing inside of your body, you're removing it. The drug is used to actually cause you to remove materials from your lungs. Here's a picture of that bladder, taking a moment so you can actually see the apical surface here. So this would be what type of membrane? This would be a stratified cuboidal epithelium, and we say that because these look like the apical surface has cuboidal cells, and it doesn't matter what the bottom ones are, but you can see it sitting on top of a basement membrane, and they transition. They transition as a result of the force of the fluid inside the bladder acting on the surface of the cells here. What I want to transition into in this last couple of minutes is how else can we use epithelial cells? Well, Mother Nature has two types of glands that can be made. One is exocrine, the other is endocrine. We start with the endocrine cells, and that's because in between these spaces around this follicular structure, a follicle is a circular-like looking structure that is made up of cells that are all adhered together. So the basement membrane, believe it or not, is over here. And the vasculature is bringing materials to it, so diffusion brings materials to these epithelial cells. Remember, they are avascular. And they're going to make something. They're going to store it inside that thyroid follicle. And when we need it, we bring it back up into the cells and we put it inside the bloodstream. So Mother Nature has the ability, whoops, what did I just do there? Has the ability to actually make materials and store them inside small areas. And when she does that, you can see here that, we've fulfilled all the rules. The basement membrane is here. Look, we've got a basement membrane, we've got blood vessels, we can bring stuff there and it's going to function properly. We also have exocrine glands. These are sort of sweat glands or sediferous glands, or the glands that make up the earwax. And just think about saliva for just a second. What's happening is they're moving materials to the surface or a lumen of a hollow organ. And if you think about your stomach, your digestive system is essentially a hollow organ, and you're pumping materials into that. And what Mother Nature does, following those same rules, materials travel into the actual dermis. They diffuse up towards the epithelia, but inside that dermis, they're making materials, and it's going to be secreted onto the surface of the cell. Think about the last time you actually went for a run, how much sweat actually ended up on the surface of your skin. Where did that come from? It came from a tubular structure. Those are all exocrine glands. So we can actually classify them in terms of their shape. Don't worry so much about, do you remember if they're simple glands or compound glands or acinar glands? That's not what's important. What is important is, do you see the types of exocrine gland? You can see that this is a holocrine or an apocrine or a merocrine, where the merocrine looks like that normal secretory pathway that we studied in chapter 3. Whereas apocrine is you literally pinch off the apical surface of the cell. Holocrine is the entire cell becomes a secretory product. Literally, the cell dies as it's releasing everything inside of whatever the surface is going to be, right? Okay. So this is the time to actually transition into connective tissue. So I'm going to pause here. I'm going to come up with some questions for that second part, and we'll get back to work.